Hello everyone, my name is Joana Carvalho. I am a postdoctoral researcher at the preclinical MRI group of Champalimau Foundation, and in the next minutes I would like to show you how can we map the influence of visual experience during cortical reorganization in adulthood. In human and non-human primates, fMRI experiments harness complex visual stimuli setups to perform visual field mapping. However, such mappings are not yet possible in the rodent visual pathway, where in-depth biological insights can be obtained due to manipulations of vision, vis-a-vis plasticity, and orthogonal validation. In this study, we developed the first of its kind setup to perform complex visual stimulation inside rodent preclinical scanners. Combining this setup with specially designed stimuli such as spatial frequency tuning curves and retinotopic mapping, we were able to investigate how cortical reorganization and function differs between dark-reared or visually deprived animals and healthy controls. We investigated that in 20 adult long-event rats scanned under medetomidine sedation at 4 different time points. At the first moment of light exposure, 1 week, 17 and 27 days after light exposure. Using this unique setup, we measured robust pole activation upon complex visual stimulation in the rat visual pathway. You can note that activation is circumscribed to the regions of interest, such as the visual cortex, the LGN, and the superior colliculus, and that across stimulus type, the activation patterns are very similar. Remarkably, we mapped population receptive field properties across the entire visual pathway at an unprecedented resolution and for the first time in rodents. You can appreciate that the entire visual pathway is retinotopically organized, as you can see by the nice gradient of colors encoding the polar angle moving from left to right visual field. In addition, by aggregating the receptive field properties that we obtain per voxel across the entire ROI, we can obtain a representation of the visual field. You can appreciate that the obtained visual field coverage mapped what we expect by the positioning of the animal in the bed, which should have a higher coverage of the superior visual field. In addition to the population receptive field position, we also obtain a population receptive field size, and we examined how this varies across cortical layers. We saw that the population receptive field size varies with cortical thickness, being the largest at the most superficial and the most deepest layers, and the smallest at layer 4. You can see that the higher variability at layer 6 is most likely driven by the fact that we are using a surface coil. And because we can obtain these receptive field maps per voxel, we can see how nicely the receptive field size varies across cortical depth within the same colon. So here we can see that it goes from larger sizes to smaller to then larger again in deeper layers. Furthermore, using the spatial frequency tuning stimuli, we mapped the spatial frequency selectivity across the entire rat visual pathway, and we can appreciate that all the ROIs showed to have a bandpass filter behavior towards low spatial frequencies, and that the optimal spatial frequency actually varies between ROIs, and that the optimal spatial frequency values are actually very well in agreement with what had been reported in the literature measured via calcium or electrophysiology, which is here denoted in yellow. So you can see that the peak of the green curve, so it's our derived tuning curves based on the bold responses to the stimuli, is very well aligned with the yellow band reporting the values via electrophysiology in the literature. Now moving to the visual deprivation results, we saw that visual deprivation boosts the neuronal responses and it results in faster onset times in the visual cortex. As you can see here on the T-maps, that we have really a boost in activation in our visual cortex, and it's actually 3 times the amplitude as the healthy controls. You can see that also at the later time points, such as 7 days, 17 and 27 days after light exposure, the bulb activation returns to baseline levels, and by baseline I mean the level of the controls, and this is not significant anymore. Importantly, we looked at how the population receptive field properties varied between healthy controlled and visually deprived animals at multiple time points. And we saw that at the first moment of visual exposure, t equals 0, while the healthy controls have a highly retinotopically organized visual pathway, the one of the visually deprived animals is much more disorganized, lacking this retinotopic structure, which is evident by the curve of variation of the receptive field properties as function of the distance in between voxels. As the animals are more progressively exposed to light, we can see that the retinotopic structure of the dark-reared animals becomes more and more retinotopically organized, and this is also expressed by the fact that the curves becomes much more similar than at time point 0. Next, we investigated whether the progressive retinotopic reorganization is accompanied by a shrinkage of the population receptive field properties, and we see that at T0 the PRF size in the visual cortex and superior colliculus of the dark-reared animals is much larger than of the healthy controls. We also see that for all the ROIs between the first moment in light exposure and 1 week of light exposure, there is a significant shrinkage of the receptive field size in the visually deprived animals that then is maintained at the baseline level. And this is very well depicted actually in these representations of how the receptive fields evolve across time. And because of this reason, we think actually that the shrinkage of the receptive field is the motor of the reorganization of the visual pathway. A similar mechanism is also present in the specialization of the spatial frequency turning curves. While at time point 0, the visually deprived animals here in orange show to have a broad turning curve without a specific peak, contrasting with the healthy controls in green. 1 week after light exposure, we can appreciate that visually deprived animals have a shrinkage of their spatial frequency turning curves, and the peak becomes aligned with the one of the healthy controls, which is actually depicted in these graphs. To sum up, I have showed you that the rat visual system is retinotopically organized and that in the visual cortex the receptive field size varies with cortical depth. I also have showed you that we mapped for the first time the spatial frequency selectivity non-invasively in the rat visual pathway. We showed you as well that visual deprivation boosts neuronal responses and results in faster onset times in the visual cortex, and it leads to differential bulb activation patterns in the superior colliculus. The adult rat brain is also highly plastic, and the visual deprivation delays the maturation of the visual system, in particular the retinotopic organization and spatial frequency selectivity of the visual pathway, and that light exposure, even during adulthood, post-critical period, can clearly promote the specialization of the visual system in visually deprived animals. Thank you.